Elabscience One-step TUNEL Assay Kit is a highly sensitive, fast, and simple method for cell apoptosis detection. Positive, negative, test. Preparation of cell meal. Remove the liquid from the culture plate. Immerse cell cover slips in 4% paraformaldehyde. Incubate at 4 degrees for 1 to 2 hours. Remove 4% paraformaldehyde. Wash the cover slip with PBS for 3 times, 5 minutes each time. Remove PBS. Immerse cover slips in 0.2% Triton X100. Incubate at 37 degrees for 10 minutes. Remove 0.2 Triton X100. Wash the cover slip with PBS for 3 times, 5 minutes each time. Preparation of positive control. Use double distilled water to dilute 10 times DNase1 buffer into 1 time DNase1 buffer working solution in the ratio of 1 to 9. Mix. Transfer the cover slip to the slide. Add DNase1 buffer working solution to cover positive sample. Incubate at room temperature for 5 minutes. Use 1 time DNase1 buffer working solution to dilute DNase1 to DNase1 working solution in the ratio of 1 to 99. Mix. Remove DNase1 buffer. Add DNase1 working solution to cover positive sample. Incubate at 37 degrees for 20 minutes. Transfer the cover slip to the culture plate. Wash the cover slip with PBS for 3 times, 5 minutes each time. Labeling and nuclear staining. Transfer the cover slip to the slide. Add TDT equilibration buffer to cover each sample. Incubate at 37 degrees for 10 to 30 minutes. Prepare labeling working solution of positive control and experimental group. Prepare labeling working solution of negative control. Mix. Remove TDT equilibration buffer. Add labeling working solution to cover each sample. Incubate at 37 degrees for 60 minutes in humidified chamber. Transfer the cover slip to the culture plate. Wash the slide with PBS for 3 times, 5 minutes each time. Use PBS to dilute 25 times DAPI into 1 time DAPI in a ratio of 1 to 24. Mix. Transfer the cover slip to the slide. Add DAPI working solution to cover each sample. Incubate at room temperature for 5 minutes in the dark. Transfer the cover slip to the culture plate. Wash the slide with PBS for 4 times, 5 minutes each time. Transfer the cover slip to the slide. Add anti-fluorescence quenching agent to the slide. Place the cover slip upside down on the slide. Analyze sample. Immediately analyze samples under a fluorescence microscope.